Hi everyone, I'm Evelyn Beaury, and I'm going to be talking about a macroscale analysis of the biotic resistance hypothesis, which is the effect that native biodiversity has on non-native plant invasions. Invasions follow a common process in which a non-native species is introduced to a new ecosystem. It spreads rapidly, and once it reaches high densities, it can have negative impacts to native biodiversity and ecosystem services. Because the damage non-native species can cause when they reach these later stages of the process, it's important that we understand what factors affect colonization and establishment so that we can develop management strategies to limit the number of species that go on to be invasive. One of the primary and more controversial hypotheses for what affects a non-native species' ability to establish is biotic resistance. And this is the idea that communities with more native species occupy more niches, and it's harder for incoming non-native species to establish. So under this hypothesis, we would expect that species-rich native communities have lower numbers of non-native species, and these communities have higher resistance to invasions. I called this one of the more controversial invasion hypotheses. That's because previous studies of biotic resistance have mixed results. So on one hand, there are experimental studies, which typically occur at smaller scales in which researchers are manipulating native biodiversity and monitoring for what non-native species establish. And these studies find pretty consistent support for biotic resistance. On the other hand, are observational studies in which researchers are compiling plant surveys across wider geographic areas. And these studies not only find no evidence for resistance, but they actually find the opposite trend, where communities with high numbers of native species also have high numbers of non-native species. One reason for this disagreement may be that observational studies, and typically those that occur at the macro scale, may fail to control for spatial variation in species richness across sampling areas. For example, if you sample across a single study site or habitat type, you might capture minor variation in species richness. But if you sample across a more heterogeneous landscape or across biomes, the species numbers are going to vary with habitat. So you might see high numbers of native and non-native species in areas with high resource availability and low numbers of species in habitats with fewer available niches. On a plot, you might expect something like this, where you throw all your data together and you see a positive trend between native and non-native richness. But in reality, these differences are driven by differences in habitat, not by a lack of biotic resistance. To retest the biotic resistance hypothesis at the macro scale, we took a modeling approach that controlled or tried to control for the natural variation in species richness that may have caused the disagreement between past experimental and observational studies. So I don't have a lot of time to get into the nitty-gritty of the methods, but I will quickly note kind of the key changes that we made. First being that we reused a really awesome data set from the National Park Service that surveyed plant communities at the plot level across a really wide diversity of habitats. Second, we used a binomial model to predict a likelihood of non-native occurrence. And this likelihood of occurrence assumed that each community had a fixed number of niches represented by total species richness, and non-native species occupy a proportion of these niches. So using this proportion allowed us to analyze species richness relative to the different habitat types that were sampled by the Park Service. And lastly, we used a model that included several spatial environmental predictor variables to try and capture differences in habitat type that could have been causing that variation in species richness that kind of distracted from the biotic resistance relationship in past studies. Using this approach, we found consistent evidence for the biotic resistance hypothesis across the 28 different habitat types sampled by the National Park Service. So here, each plot represents a habitat type, and across the board, we found that non-native occurrence significantly decreased with increasing native richness. We also found that biotic resistance varied with habitat type. So here are all the predictive relationships from that last slide, and you can see that they vary around this overall trend shown in black. So one example being that forests had significantly lower likelihood of non-native occurrence compared to herbaceous communities. So forests are these darker green lines here, and herbaceous communities are the lighter green lines here. We think that under the biotic resistance hypothesis, this reflects differences in niche availability in forests versus herbaceous communities. So forests are typically dominated by older growth species, whereas herbaceous communities have more seasonal turnover in species. So there might be more kind of seasonal niche openings for non-native species to establish in herbaceous communities compared to forests. We also found that proximity to human activity affected non-native occurrence. So human activity is known to cause disturbances that increase the introduction of non-native species and kind of facilitate the spread of invasions. So here we found that non-native occurrence was higher in areas of high human impact, and that's these darker red lines here, with one example being the Golden Gate Recreation Area, which is a site that had high likelihood of non-native occurrence. It's directly adjacent to a really urban area, versus something like the New River Gorge Park in West Virginia, which is really remote and had lower numbers of non-native species. So altogether, these results provide pretty widespread support for the biotic resistance hypothesis, with a few important implications I'd like to point out. So first, since this is a session on macroecology, I want to say that the scale of the data and the modeling approach are really important, especially when you have data across a really wide diversity of habitats. So we found that by changing the probability distribution and adding key predictor variables to the model allowed us to capture spatial heterogeneity that was likely skewing some of the results of past macroscale studies of biotic resistance. And second, I want to make the point that conserving native biodiversity is an important way to reduce non-native species occurrence. And by reducing that occurrence, you reduce the pool of species that could potentially spread and become invasive with negative impacts. Obviously, we knew native biodiversity conservation was important, but this study really provides evidence that plant communities benefit from management strategies that maintain high levels of species richness. So some examples would be planning active restoration projects rather than passive restoration, or monitoring for invasive species establishment post removal when you lower kind of the number of occupied niches. And this is especially true in habitats where biotic resistance is weaker. So with that, thank you for listening, and a quick thank you to my co-authors and other collaborators at UMass. I put my email address and Twitter account here, so feel free to contact me with any questions. And our next steps are to use the abundance data from the National Park Service to study other macroecological and invasion hypotheses. So if you want to talk about that, please let me know, and I'd be happy to discuss that as well. Thank you.